DNA forms the genetic material in all living organisms. In the prokaryotic cells, DNA occurs in the cytoplasm. In the eukaryotic cells, it is confined largely to the nucleus and forms the main component of chromosomes. A small amount of DNA is found in the cytoplasm in the organelles like mitochondria and plastids. It is called extranuclear DNA. DNA is a polynucleotide of adenine, guanine, cytosine and thymine. The structure of the DNA molecule and the arrangement of nitrogenous bases in the two polynucleotide chains is very well explained by the double helix model proposed by Watson and Crick in 1953.
The double helix model of DNA, deoxyribonucleic acid, consists of two intertwined strands. These strands are made up of nucleotides, which themselves consist of three component parts: a sugar group, a phosphate group, and a base. The sugar and phosphate groups combined form the repeating backbone of the DNA strands. There are four different bases that can potentially be attached to the sugar group: adenine, thymine, guanine and cytosine. Given the designations A, T, G and C, the bases are what allows the two strands of DNA to hold together. Strong intermolecular forces called hydrogen bonds between the bases on adjacent strands are responsible for this. Because of the structures of the different bases, adenine, A, always forms hydrogen bonds with thymine, T, whilst guanine, G, always forms hydrogen bonds with cytosine, C. In human DNA, on average there are 150 million base pairs in a single molecule.
According to the double helix model, DNA molecule shows the following salient features. The two polynucleotide chains are coiled around each other like a spiral staircase, double helix. The cross rungs, steps, are formed by the nitrogen bases while phosphates and sugars form the uprights. The two polynucleotide chains run in opposite directions, antiparallel. One chain runs in the 3-5 direction, while for the other chain 3-5 direction is opposite. The average distance between the two chains is 20 angstrom. One full turn of the helix, called gyre, measures 34 angstrom in length. The distance between two successive sugar molecules is 3.4 angstrom. Thus each gyre accommodates 10 nucleotides. The nitrogen bases of the two opposite chains exhibit highly specific base pairing. A purine in one chain always pairs with a pyrimidine in the opposite chain. Among purines, adenine, A, pairs only with the pyrimidine thymine, T, and vice versa. Similarly, guanine, G, pairs only with cytosine, C, and vice versa. There are 2 weak hydrogen bonds between A and T or T and A. There are 3 weak hydrogen bonds between G and C or C and G. The total amount of purines is equal to the total amount of pyrimidines, A plus G, equals C plus T. The two polynucleotide chains of DNA molecule are not identical to each other but complementary to each other.